Firstly, the test drug needs to be added. In the 96 well plate, the labeling is done accordingly. The first 3 wells constitute control, which are the untreated wells, and the next 3 wells constitute the solvent control, to which we'll be adding DMSO, that is vehicle control. The next 3 is positive control, to which Triton X100 is added. From A10 to B9, various concentrations of quercetin will be added in increasing order. 0.1 micromolar, 1 micromolar, 10 micromolar, and 100 micromolar. Each and every treatment here is done in triplicate to have concordant results. So right now, we'll be adding solvent control and the various concentrations of drug. And after 24 hours, positive control, that is Triton X100, will be added to the respective wells. Since we're using a detergent here, if we keep it for a very long time, the cells will be completely lysed. So we'll be adding Triton X only 20 minutes before the start of the experiment. Solvent control is added first from A4 to A6. Next, we'll add various concentrations of the test drug as a single shot. 2 microliters of the test drug is added according to the concentrations that have already been mentioned. 0.1 from A10 to A12. Next, 1 micromolar from B1 to B3. Ten micromolar from B4 to B6. Lastly, 100 micromolar from B7 to B9. Now, the plate will be incubated in the CO2 incubator for a period of 24 hours. So it's kept in the incubator. After 24 hours of incubation, the plate is now removed from the incubator. So now we've added solvent control and treated cells. So we'll be adding positive control, that is Triton X100. And we'll be adding 2 microliters of that to the positive control wells. As mentioned earlier, Triton X100 will be added later on because it's a detergent and might completely lyse the cells. Now this will be incubated for a period of 20 minutes. After 20 minutes of treatment with positive control, we will be starting the assay. The contents of the well should be discarded and PBS must be added to it. And this PBS should be added to the sides without disturbing the cells. So the contents are discarded and PBS is taken. In B12, only PBS is added. It does not contain any cells, so it will be serving as a blank. And this is incubated for a period of 10 minutes. After incubation, we will be adding MTT dye to all the wells except the blank. Since MTT is light sensitive, we have to switch off the light and add 20 microliters to all the wells. So after the addition of MTT dye, we'll cover it with aluminium foil and incubate it in the incubator for next 3 hours. We cover it with a foil to avoid exposure to light. So it's kept in the incubator. And after 3 hours, it's taken out from the CO2 incubator. Now we unwrap the foil and the contents of the wells are discarded carefully without disturbing the cell monolayer. One by one, the contents of all the wells are discarded as shown. So now the contents have been discarded. Next, solubilization buffer containing DMSO is added and the plate is incubated for another 2 hours in the dark. Make sure to wrap it with aluminum foil again. After 2 hours of incubation, finally the absorbance is read on the spectrophotometer as shown. We use 570 nanometer wavelength. Remove the aluminium foil and place the 96 well on the plate reader as shown. Click on the Start option. And type out the required wavelength. And so here we have our respective readings. And this is the 96 well plate.